and do something fairly drastic in terms of the scientific approach, because the experience here is truly daunting. This is a paper from Nature Reviews Drug Discovery, just published a couple months ago. These authors basically tallied up what the cost was in terms of drug development since 1950. And using inflationary corrections, they plotted here the number of drugs per $1 billion of R&D spending, and they plotted this out on a log scale. And you can see it looks really troubling here. It almost looks like some sort of law is at work here. And it's a law that's going the wrong direction. Now down to sort of less than one drug per $1 billion, where back in 1950, goodness, there was more like 30 or 40 in inflationary corrected dollars of that same thing. So what's going on? The authors, somewhat tongue-in-cheek, decided maybe this needed a name. It sort of looks like a law, so they called it Eroom's Law, which, if you notice, is Moore's Law written backwards, because this is going backwards. So what's the problem? Well, the engineers in the room would probably say, that's your pipeline? That's the best you can do? What's going on here? Why all the failures? Why the slow progress? What could you do to choose your targets better to begin with? How could you figure out how to fail early instead of late? All of those are critical issues. And science has come along in interesting ways in the last few years to enable us to tackle some of those questions in truly innovative fashions instead of continuing to do things the way we have, which clearly from this diagram is not working so well. So this really was the motivation for us here at NIH, after much consultation through many different venues, particularly the Scientific Management Review Board and consultations with the private sector through my advisory committee to the director. We established the National Center for Advancing Translational Sciences just last December 23rd. And it is not intended at any sense to be the place where all translation happens at NIH, not at all. It's intended to be a hub, a catalyst, to look at actual bottlenecks in this pipeline as themselves scientific and engineering problems, not specifically attached to any disease or any specific small molecule or diagnostic or device project, but looking systematically at the way the whole pipeline works, or in many cases does not work. So the NCATS has already gotten itself deeply engaged in a variety of projects, one of which fits rather nicely with one of the examples that Roderick showed you, is to try to utilize what we're learning about tissue engineering to be able to do a better job of predicting whether a drug is safe or not before you give it to a patient in a phase 1 trial. And this has led to an unprecedented collaboration between NIH, DARPA, the Defense Advanced Research Projects Agency, who are pretty good at wild and crazy engineering ideas, and FDA. And collectively here, we're putting $140 million into this over 5 years. And the goal is to develop a chip loaded up with as many as 10 different cell types, human cell types, in a fashion that is as much as possible representative of what happens in vivo. So as you saw in the video a few minutes ago about the human liver that was being reimplanted in mouse, in this case, we'd want to have human liver in a 3-dimensional organoid on this biochip, as well as cells representing heart, kidney, brain, and so on, the places where you most would be interested in seeing a signature of toxicity if that was likely to be a problem. And of course, we need to wire this chip up with a lot of different outputs in order to find out everything we can about what happens to cells when they're exposed to that new test compound. So that probably means looking at gene expression, at metabolomics, at proteomics, and so on. And here is where we hope the science that NIH is pretty good at and the science that DARPA is pretty good at can come together. So there is, between our two agencies now, efforts to put together a consortium. And this will be an interesting cultural experience as well, bringing together staffs of NIH and DARPA to work on this. Awards are anticipated imminently next month, where we hope to put together some of the best ideas about tissue engineering, biology, toxicology, and so on, to create a pathway towards generating such a biochip. Obviously, the goal will be to assemble this in a fashion where you can then test it with compounds where you know the answer. That they are safe or they're not safe, and then develop the idea about what a signature would look like that would give you confidence that it was reasonable to go ahead with a phase 1 trial or not reasonable. If this works, presumably it should be much faster and cheaper than the slow, expensive, and not very reliable animal testing, which is the current way in which you have to generate evidence that your compound is safe enough to get an IND from FDA. So FDA's involvement here is critical, because if this begins to work, we would not want to see this as an add-on, as an additional thing that you have to do to test for safety, but rather as a substitute for the methods that are currently used. And that is an example of the kinds of things that NCATS aims to support. And you've already seen one part of this, the lung on the chip. I won't go through it again because you already even saw part of this video and saw it much nicer because it had some narrative to go with it. I will show you another video quickly. This is one I showed yesterday to the House Energy and Commerce Committee, and it is work supported by NIBIB, the work of John Donohue and others, which is basically then developing this interface between the brain of individuals who have quadriplegia and allowing them, through the process of connecting that interface with their own brain function, to learn how to move a robotic arm simply by the thought process that the subject is undergoing. I must tell you, the members of the Energy and Commerce Committee were quite riveted by looking at this. Here is the example where the patient in this case is attempting by her thoughts to pick up this canister, which has coffee in it, and see whether she can bring it to her mouth and take a sip of her own coffee. All of this being accomplished, as you can see, by the box mounted on the top of her head that allows her thoughts to be translated into that robotic arm. And I think you can see by the smile on the face of the patient and the investigator that this was a pretty good day, that this really was a remarkable moment. Well, it's great to have all of these capabilities, but we certainly need not only to have great science and great ideas, we need to have the talent of the individuals to pursue that. NIH is intensely interested in encouraging the most innovative ideas to come forward, and worried that in the current climate, where our budgets are very tight, that peer review sometimes can be a little conservative in terms of not taking as many risks because there's such great solid science in front of them as well. And one of the things we have done in order to try to encourage that kind of innovative approach is a series of special programs, the three you see here, all supported by the Common Fund, that aim at high-risk, high-reward research. The Early Independence Awards, which we've just started, aim to take the most talented graduate students and take them directly from the PhD degree to independence, skipping the postdoc and giving them the chance to show their stuff while they are still in that oftentimes most creative phase, and not in a circumstance where they are following other people's guidance but allowed to put their own ideas forward. We just made 10 of those awards in this first year. The best day I think I had in the last 6 months was coming to listen to those 10 grantees talk about what they were going to do. A truly remarkable and inspiring group of fearless young investigators who are aiming to do things that were quite bold indeed. The new Innovator Award also aims to try to bring new investigators to NIH who've not previously received grants from us, not requiring nearly as much in the way of preliminary data, but demanding that what they put forward has to be a groundbreaking kind of approach. Otherwise, they can't be evaluated as part of this program. The Transformative R01, and now called the Transformative Research Award because it isn't really quite so much like an R01, also has to be transformative but can support large teams and has less in the way of budget limits than many of the other kinds of formats that people are used to. And the Pioneer Award, which has now been around for more than 5 years, which gives an investigator the chance to write a relatively brief proposal with some bold ideas and then to be funded for a 5-year period as an investigator, and basically allowing them to move in the direction that makes the most sense scientifically, not particularly tied too tightly to original plan, very much like a Howard Hughes position. And those also have turned out to be very much productive parts of our portfolio. As an example, I'll talk about an individual who's actually received a Pioneer Award and more recently a Transformative R01, and who is a grantee of NIBIB. And this is Sunny Shi. This is an investigator at MIT who's a chemist who has actually applied some very innovative approaches to do single molecule visualization in single cells, an area of intense interest. In fact, we're about to start a new Common Fund effort in single cell biology. Sonny Shi, using his skills as a chemist, has been able to come up with systems that allowed you to look at expression of single protein molecules. What you're looking at here from a publication in 2006, those yellow dots there are single proteins which are marked by a version of GFP called Venus. And basically, this is a circumstance where they've created a transgene in this bacterial system that is only rarely expressed because it's actually repressed by the lack of repressor. But occasionally, one copy of the RNA gets read off anyway. And that one copy then is used to make 3 or 4 proteins which appear as bright dots in that particular bacterium. And that is kind of fun to watch. Here you see the solid static version. We can run a little movie here. Now watch the yellow dots as they begin to appear. They're just sort of firing off there as an RNA was transcribed, and then proteins are briefly made from that RNA in that same cell. That was fun. Let's watch that again. See that yellow dot now will pop right up there. You can see, must have made an RNA there. Now maybe 4 or 5 copies of the protein being generated from that transcript. And teaching us a lot of interesting things. It's obviously nice eye candy, but it's also teaching us a lot about biology. He has gone on with a colleague at MIT, Xiaowei Zhuang, who's been also a very creative individual in terms of figuring out how to see things at a resolution that you thought you couldn't do with light microscopy using this technique called STORM. And here is just one image here again of an E. coli system looking at a nucleoid-associated protein called HNS. This is what this would look like in phase contrast. Fluorescence of a conventional sort would look like that. Look at the level of resolution showing where that protein is in these individual bacterial cells. So lots of exciting technology being supported through these grant mechanisms, the Pioneer Awards and the Transformative R01s. I wanted also to mention something that NIBIB is doing, which I think is kind of exciting, bringing young scientists at undergraduate level into the realm of competing with each other for this debut challenge. And this is basically NIBIB's opportunity for people to compete for prizes. The deadline for receipt of the applications was June 2nd, so presumably there will be some announcements before too long. Diagnostic devices is one approach, therapeutic another, or technology to aid underserved populations in low-income circumstances or individuals with disabilities. So very much an innovative way to try to drum up excitement in undergraduates. They have to be submitted as teams, and as you can see, there is money involved, which tends to get the attention of undergraduates. Finally, there is this issue about how we need, in the current climate, to be sure that we're making our case for the value of what biomedical research is doing, not only for the future of human health, but for the economy. And again, I think as all of us are called upon to defend the investment of taxpayer dollars in this activity, we should be prepared to say why this is a good investment. We need to make that case because we are not at the present time enjoying a particularly favorable environment for the support of biomedical research through NIH. You can see here in this series of bars, the purple bars tell you what the appropriations have been to NIH over the course of the last 13 or 15 years. You can see the doubling that happened between 98 and 2003, which was a wonderful opportunity for growth and for new investigators to come into the field. But you can also see that we flattened off pretty badly after that. And if you look at the yellow bars, which take account of inflation, we are actually losing ground and have been since 2003. Our purchasing power now is down by about 20% over what we could do 9 years ago. And obviously with the current focus, and appropriately so, on our difficult financial circumstance and deficits, there is certainly no indication that this is going to get any better, and presumably it could even get a lot worse, especially if those sequesters that are being talked about happen to kick in on January 2nd of next year. If